Okay, so people are trickling in, so we can get started. Thanks everybody, for especially those of you who are also up very early this morning. I hope you have a nice hot cup of coffee with you. I want to thank you for joining us and for our guest as well. Before we get started, I would like to acknowledge that my home office where I am today and my workplace at McMaster are located on the traditional lands of the Mississauga and Haudenosaunee Nation, and within the Dish with One Spoon Wampum Agreement and the lands protected therein. And just to emphasize that our responsibility as settlers on this land is to respect the land and its traditional owners, and to do our part to mend relationships, treaties, and promises that have been strained or broken. I think this is about our fifth speaker in this series, so I would like to welcome Dr. Sophia Ananiadou, and I might have said that wrong, but Sophia, you can correct me, please. And she's a professor of computer science and the director of the UK National Centre for Text Mining at the University of Manchester, and also a fellow of the Alan Turing Institute in London. And today, Sophia is going to be talking about automating systematic reviews using text mining, but she's also mentioned she'll be talking about text summarization. So there might be a bit of technical thrown in with practical for today. So thank you, Sophia, and I'll let you take it away.
Thank you. Just if possible, if everybody could mute there, because otherwise I'm going to be interrupted. Thank you. So I'm going to share my screen now. So let me do this properly. Okay. Can you all see my screen?
Yes.
Yes. Yeah.
Great. So I kept it a bit natural language processing applications, because what I'm going to be talking is work and text mining NLP methods that we have used for systematic reviews. Some that have been embedded into systems we have developed, some are in the process of being embedded. So you all know the process. I'll start directly by the NLP for systematic reviews. Obviously, you have a search strategy that we all know, and there are different ways of doing search. So I will mention a bit about more specific methods we're using to do search, like, for instance, term-based, but natural name entity recognition. And then the next step is to filter all those not ineligible studies. And by that, we can also do data extraction, find the relevant snippets of information in text, which actually answer a research question. This in our field, we're calling it question answering. And the filtering of ineligible studies, we can use different information retrieval techniques, but also different types of ranking methods and prioritization. The last part is the synthesis, the summarization of findings. And in this, I'll focus a lot about synthesis, because in our speak, in natural language processing, this is mapping to what we call summarization. And there are very many ways of doing summarization. So I would spend a lot of time about techniques supervised and unsupervised, but also how, by injecting knowledge into the pre-trained language models we're developing, we can have a more informed summarization. So the challenge is, obviously, you already know that if you're doing systematic reviews, it's time consuming. You know how much the process, how slow it is. And we all know that if you have a rapidly evolving literature, as we do, that the process can be very time consuming. So natural language processing or text mining, but I prefer to use NLP because it's a bit broader, accelerates all this development of systematic reviews. First of all, it improves search, and I'll talk a bit about that. then semi-automates the citation screening, which is part of the system we have developed, the robot analyst, and then the summarization, which would be a big focus of my talk. So by the search strategy, we all know that we have to increase to maximize the sensitivity and balance sensitivity and specificity. So for sensitivity, we want to have all relevant studies in the review. And ways of doing that is using techniques such as query expansion. And by query expansion are finding automatically synonyms or term variants or related terms, like, for instance, impaired vision or loss of vision. Now, these methods, this way of increasing sensitivity, they all correspond to methods in natural language processing, from query expansion, for what we call normalization, also using distributional method semantics to find related terms, etc., etc. But also the sensitivity, the technical terms and other words, they have the variation, they include acronyms, different morphological variants, and all these actually methods support the increased sensitivity of the search. Now, the specificity is really, we disambiguate. So we want, if we're studying, basically, we're looking at a study about proteins in biology. Cat, it's a protein, but it could be also an animal in a different collection of documents. So we want also to disambiguate these, for instance, acronyms. And also we want to be able to, for instance, GAD might refer to either generalized anxiety disorder or glutamic acid decarboxylase. So those are methods that allow us to balance, to maximize both. And we also can use queries. We can query the document collection, this is the question answering, using what we call subjects, basically verb-object templates, rather than keywords. So to sum up, actually, in that kind of literature search, we want to be able to improve search mechanisms by increasing specificity and sensitivity. So I'll start with the name entity. So this is an example of what we call in our field name entities. And then by name entities are semantic, basically, hats on top of words, as to put it, of strings. So EGTA is a chemical, alpha AR is a protein. So we have a name entity is a word or a group of words that constitute a proper name. And the entity type is the semantic category that belongs to. And very often now in name entity, we do spans. So we do recognition based on text spans. And by that, we're looking at all the words that are included in this entity. So for instance, it could be alpha AR or 50, so numbers, etc. So the input here is really a textual snippet, a sentence, and the output is a label for each token in the sentence. Typically, the name entities are predefined. So we know when we start developing the recognizers, typically users are telling us we want to find, for instance, proteins or genes or disease names, etc. And the performance in those systems are pretty good. It's almost human level performance in NER. So the usefulness is that this part is very important for other tasks. And those other tasks could be, for instance, to do search, to do question answering based on those name entities, but also subsequent tasks such as extracting relations between the entities. So in relations between, for instance, genes or proteins and, or populations. Now, it's not, of course, so straightforward because the biomedical text, as I said before, is not only very ambiguous, as you see with the acronyms, but also it has a lot what we call nested entities, which I'll explain later. And to do that in our field, because we do the whole process fully automatically, we need to use contextual information. And this could be at sentence level and very often at document level. So in general, as we're going to have a granularity of name entities, it becomes the topic very challenging. Mainly, if you're using techniques like supervised techniques, you need to have labeled data. And if you don't have enough labeled data, this is problematic. All other ways of going around it. So in case you don't know anything about natural language processing, which I hope some of you have. So initially, I mentioned all that. There's a whole, progress in our field. We started with rule-based methods. I talked about the acronyms. People in the past were doing, for instance, acronyms by writing rules. Then for a long time, we have feature engineering methods, I would say for about 20 years. And those are really using, different types of features or lexica, etc. And then we're going to the deep learning techniques, which I'm not going to describe here. But those are especially BILSTMs, a combination of BILSTMs and CRFs have been very popular in name entity recognition. And now everybody, more or less, everybody is doing deep learning, transfer learning. We're using different BERT models. And I'll explain why I'm mentioning here BlueBERT and BioBERT. So the more domain adapted is the pre-trained language model, the better for the performance of a specific field. So the nested entities, and I'll just give you a small example here. If you look at the sentences, he was given albuterol nebs for presumed narcotic-induced respiratory distress. So here we have a narcotic-induced respiratory distress. First of all, it has narcotic, which is a drug, embedded into two other things, adverse drug events and reason. Also, mouse interleukin-2 receptor alpha gene expression, protein is interleukin-2, but this is also embedded inside the longer string, which is receptor alpha. So we see then that when we want to automatically recognize entities, we need to be able to understand that entities that can share the same text span, but they have different types. So there is polysemy here. And so we need to take into account to create models that actually are able to automatically recognize this. Now, I know it doesn't sound great, this one, if you're not into deep learning, but this is, you can just forget it if you don't care. But for the people who do, it's basically a kind of a model based on, to understand, to detect nested NERs from any type of text. The interesting part of this is that we want to make our systems as independent from external resources, because you can extract name entities using dictionaries, but then you're very much depending on the dictionary. So you can use it, of course, but it's best to have systems where it's quite general. So it's basically a kind of sequential stack of what we call flat NER layers that detect nested entities in an end-to-end manner. So, so each specific NER layer consists of a single LSTM layer and then a CRF. So if we look at the sentence here, the mouse interleukin receptor alpha gene expression, we have protein and we have DNA. So first we stack one flat NER layer to identify the inner entity, which is interleukin-2. And then this is used as input to identify the outer entity, which is interleukin-2 receptor alpha gene expression. So this is basically a stack, if you want, reuses a flat NER layers and then stops if the moment we don't find any more. So I'll just go then to say a bit more about why this is important for PICO elements. And this is actually I assume your interest in systematic reviews. I don't have to explain to you about PICO elements or PICOs or whatever. So those are obviously very important elements in systematic reviews. But as you can see here from this sentence, we have the same phenomenon of nested entities. So we have populations, intervention, the population is a bit longer, etc. So methods like the ones I said before are very important, not only for identifying the specific entities, but also to work to be used for other tasks in the development of systematic reviews. Our work, we applied our system on nested NER on a PICO corpus, which has been developed by the Ni et al. Group. Those are 5,000 abstracts of medical articles, which describe clinical randomized trials. And as we can see here, we have a quite defined grain. For instance, intervention, comparator, educational, pharmacological, surgical, and different types of outcomes. So we see the performance here. We do a token and document level because we use different types of features like document level bag of word, but you can make it a bit more complex. And the performance is quite good, the precision and recall. So F measure is the mean average of between precision and recall. It's actually quite good, if we consider the granularity. This work is a bit old now. Those things are improved a lot by using other methods. So I mentioned name entities, and clearly a search system that I've mentioned before is which we have used actually for identifying personalized treatments and clinical trials for precision medicine. This is why I'm mentioning it, is that you can predefine, in this case, we predefine the concepts, the chemicals and diseases, etc. And then we developed a search system which is applied on the whole of PubMed. So going back about the search strategies that they have to balance specificity and sensitivity. So this, if we put cut, we have about 130,000 abstracts. So it's unnecessary workload for the reviewer. But then we can have a faceted search. So we specify the semantic category of the query terms. And now we reduce the number of papers to read by almost 85%, because the previous one from all those 125 abstracts, now we have 18,000 abstracts. So this is a very easy way, by using name entities, to be able to narrow down the search. So we still extract information, but we don't have to have unnecessary filtering when we are actually doing the screening. And I mentioned also about the sensitivity and the GAD, the acronyms. Again, this could mean absolutely anything. Here we might want to find the disease. So the system again tells us the different options. It could be a gene as well. And by the selection, by disambiguating the acronym, we reduce the number again from 7,000 articles to 1,000. So we increase the specificity. Those are actually exemplars of how name entities with also incorporating acronym recognition and disambiguation can improve search. So I'll mention the robot analyst now, because these types of methods have been integrated. So we looked at, although I'm not going to spend too much time on that, just to give you an example is, of course, a web-based screen system is on our website. We make it available if people want to use it. And basically, the main contribution of the robot analyst is to combine active learning to put a priority to prioritize reviews based on relevancy feedback. So by that, we also mean that the humans, they have the ability to look at the results of the system, and then by agreeing or disagreeing, you can actually harness this type of information. This is an unsupervised method, so it's unsupervised exploration of the references. And we used different search facilities, which I don't describe here. One can use terms, but we can use topic modeling. We can use descriptive clustering before we do the active learning. And in all these cases, the human is in the loop, because by selecting the elements which are actually extracted, you can augment the prioritization process. So again, a bit of more information about how this whole thing works, what I said. So first of all, you import the references as RIS files. Then you search the collection. You have terms here, it's been pointed terms, and if you click the term, you have all these technical terms that have been extracted automatically with the numbers, how many occurrences they have. Then you can search based on publication year, etc. But then you can actually start exploring the cluster, the whole document collection with other ways, with, for instance, clustering. So here we have clusters. So if you have a big collection with very the different topics, you can use either topic modeling, or you can use descriptive clustering to group the different basically areas. So for instance, one document collection talks about cancer, or about harm or strategies and cervical cancer, etc. Then here, this is where the reviewer makes their own decisions about excluding, this is automatic, of course, and there are different confidence levels. Our relevancy feedback is based on certainty. So we call it certainty-based screening and relevancy feedback. Then you make your decisions. So here the model now is updated based on your decisions. You can see undecided many and zero included and excluded. But the moment you make your decision, this part 5 here changes. So then you say, update the predictions. So the system comes now with new predictions. And you have the ability as well to put a priority, how you view the different references. You can include, for instance, you can see the ones you are more certain, or you can look at the ones which are not certain. So there are various actually decisions that the user, the system allows to the user to do. So also active learning, I have to say, basically uses new screening decisions made by the user to improve the prioritization throughout the process. And also active learning naturally handles the imbalanced sample that we have a problem, because you include, we include here references with a substantial chance of being relevant. And this is again, our decision. Here, the relevancy prioritization I mentioned before. The reason I mentioned the PICO elements here is we try to improve the prioritization reference in the document screening by incorporating the PICO information that we have extracted automatically before from the medical abstracts. So I mentioned all that, so I won't say it before, but in this case, the prioritization in the robot analyst, we didn't focus on specific PICO elements. And here we try to improve the whole prioritization, and you can improve the prioritization process in document collection using all sorts of other basically techniques. So in this case, we developed, actually, we included PICO, name entity recognition, into the system. So again, it sounds a bit, but the workflow, the reference prioritization is basically, we do it in 3 phases. The first is the PICO recognition that I mentioned before. Then we generate features. It's a bit old, so we're talking about still features. And then abstract screening. So with the PICO model that we have from first phase 1 here, we deploy the model to the second phase. And this is actually converts an abstract to vectors, because here we're talking about vectors. Then we apply the PICO model to the collection of abstracts to get the PICO elements. Now, these elements, together with their original abstracts, are used to create vector representations for each abstract. And then you have, as I said here, the different, each vector, you can have other elements as well, a bag of word representation from the PICO elements, the title, and the abstract. So the third task is basically to screen the collections now from an unlabeled data set. So the user then labels abstracts as being relevant or irrelevant. And these descriptions then are used to train the classifier model that uses the vector representation. So the difference here is that, so the user sees the top ranked abstracts, labels them, and then this process repeats. But the prioritization is based on the PICO elements. That's the main difference. So, so this is just to summarize that, because now I'm going into summarization, I'm summarizing it. The inclusion of the PICO features improve the performance of how basically the PICO tagged segments in text are predictive features for inclusion for people who are actually for systematic reviews. And we combine the relevancy classification with PICO. And it's also because people talk a lot about interpretability in our work. It's much more interpretable because it emulates how human annotation in systematic reviews. And those annotations, of course, can be used on their own to improve search. So now I'm going to summarization, and which is the last part of screening, of synthesis. And in general, text summarization, what it is, text shortens one document or maybe documents, and typically they're long documents, and that's important, into a condensed summary. So you actually want to keep the most important information. So you have the original documents. Again, you have different vector representations. And these are doc2vec or BERT or BioBERT. So those are pre-trained language models, basically, that produce the vector representations. And then we have the selection. So you have, you can use different classifiers. For instance, in deep learning, LSTM, BLSTM. So you select the items that are important for your summary. And then you have the summary generation. Now, all these elements and the data, as I mentioned before, the PICO elements and the other elements are very important because you can create summaries which are, they don't take into account the specific name entities or the specific elements are important for us. So we want to make, as I'll explain from now on for the rest of my talk, how we can improve, make summarization more pertinent for different users. But before I do that, I want to explain to you what are the different types of text summarization. So we have 2 types. The first is, we call them extractive methods. And these perhaps are more close to those, your evidence tables, but that's to be debated, obviously. So the extractive methods select key sentences from the original documents and put them together into a summary. The abstractive methods generate now, it's like an oracle. You generate new sentences. You create a summary like a human. So this is a kind of an example from the radiology reports. The extractive here, there's no joint fusion. And then the abstractive, it actually creates a story, says normal radiographs of the left ankle. So that's a kind of very simple difference between the two. So now I know perhaps because I didn't know very well the audience, but it's very important to say that my focus now is going to be biomedical text summarization, because summarization, it's really a big topic in our field and people work in general language. But in this case, I'm focusing mostly on biomedical, which is more relevant for people who work in general and systematic reviews. So there are various techniques, and the earliest methods are more traditional machine learning methods. For instance, we use naive Bayesian models, graph-based ranking methods, such as text rank, for instance. Then we have different neural network methods, and these have been mainstream for both extractive and abstractive summarization. Now, the neural extractive methods can be classified again into unsupervised. So basically, we don't need training data, and supervised data, and supervised methods. For the unsupervised methods, the model the extractive task into kind of sentence ranking problem. So, and this is actually very common. So you rank sentences in different ways, and you generate sentence representations based on different word embeddings. And then you use any unsupervised ranking method to select the most important sentences based on these representations. Now, the supervised methods, they generate what we call an oracle summary for each document with sentences which are most similar semantically to a gold summary. So a gold, if we take a paper, for instance, an abstract of a paper, a produced paper, this we consider the gold summary. So we try to create basically sentences which are semantically similar. And these methods also adopt a kind of unsupervised sentence selection method, different such as a greedy search algorithm to generate the oracle summary. So, and now I know it sounds a bit too much, but an important part of what I'm going to say, what our community is doing. So there's no way you can do natural language processing nowadays without looking at, listening to these pre-trained language models. So the pre-trained language models are fundamental for our work. And in this case, I was looking mostly at the pre-trained language models, how they're used in biomedical summarization. And again, they have the similar framework for both extractive and abstractive tasks, but they're much more powerful than neural networks in encoding biomedical text. So the issue here is, as you will see in the PLMs in biomedical, that the general, just to say this way, the most advanced pre-trained language models, and most probably you heard about BERT or variants of BERT, are also pre-trained basically on texts which are of general language. So if you just like, for instance, book corpus or Wikipedia, but it's very challenging to directly apply these models to biomedical text. And the major issue of that is the terminologies, how we use technical terms in biomedicine. And most of these terms, they have never been used in the general or domain. And if they have been used with a different meaning. The example is, say, for instance, cat and cat. One is a protein, the other is an animal. So this, though, domain specific tokens, the terms and the multi-word terms, embed what we call the salient information, which is fundamental in understanding biomedical text. So to fill that gap, many pre-trained language models for the biomedical domain are being used, like BioBERT, the clinical BERT, the SciBERT, and they basically they further pre-train language models in the general domain with biomedical text. Now, why this is important is, as I mentioned that before, actually, I just mentioned, I didn't know I had that. So we used different, basically we used biomedical literature, which is already existing, PubMed, or some PubMed, or Stork. And there are different biomedical text summarization data sets, which actually are important for us to train our models and evaluate our systems. Some is like the CORD-19, which is about the COVID-19. The CR data sets supports the task of biomedical for lay language summarization, and I'll come back to that. So even in systematic reviews, if you're producing summaries, some summaries are too technical for people to understand. So there are ways of doing summarization, but also to simplify the text, the summary, to make it more readable for a wider audience. And then we have the randomized control trial. So there are different types of summarization data sets. So now, very briefly again, I'm going to talk the various ways of, as I said, of doing summarization. And because this work is only on biomedical, that's why I decided to introduce this to you. So as I mentioned before, pre-trained language models are quite limited in a sense in the need of having labeled training data. That's why I mentioned the previous data sets. So very often, and also because they're based on transformers, they're quite limited by their attention mechanism, and I'll come back to that. The attention mechanism is how we are dealing with long documents. So if we try to summarize many abstracts, it's okay. But if you have many long documents, so we need to be able to find ways. Typically, people truncate the documents to the tokens that BERT type of models transformers can accept, which is about 512 tokens. But this is not sufficient, and it generates lots of errors. So we are trying now to address the challenges of supervised methods by adopting a more hybrid and supervised summarization method. And for that, we use salience, which is the most important element. Apologies, I have to drink some water. So our method is a kind of two-step and supervised hybrid, we call it abstractive extractive method, generates salient textual fragments based on the queries and document titles, which actually represent sections of the document, and we use this to guide the extractive summarization step. So here we have what we call fusion methods. So we're using state-of-the-art pre-trained language models with unsupervised approaches to create summaries, which first of all, they take into account the semantics, the semantic knowledge of the transformer, because most of, that's quite important of generating meaningful summaries. And the important part is to extend to any length document, because if you cannot deal with long documents, again, your summarization method is problematic. And also very important, you don't need a corpus for training data. So it's a hybrid, supervised, abstractive, extractive, unsupervised summarization method using salience for long documents. This is called Gen compare sum. I might skip that because it's all transformers and everything, but so I'll just go through the results. Now, the experiments that we have done are on different data sets, PubMed, Archive, CORD-19, and Stork. And all those data sets consist of full article research papers and corresponding abstracts. So basically, the abstracts are quite important here to learn. In line with also other methods that we have done in the scientific literature, we use the abstracts as the target summaries. To evaluate, we use existing techniques like transformers. We use BERT sum, random, lead sum, basic, and then also we evaluate use existing methods like lex rank and text rank. So we evaluate our models both on short documents and full documents, because the length of the document is quite important. I looked at both supervised and unsupervised methods for long and short versions of all data sets. So the results of that, just to give you an idea, is that the results of the extractive summarization on PubMed, Archive, etc. We are looking, as I said, at different, both short and we see that our system outperforms other methods on both long and short documents. And we have different metrics, which again, if you're not in the domain, and I'll mention that the evaluation a bit later, those are metrics that we are using for automatic methods for extractive summarization. And we also compare this with different generative models, which either generate sentences or document. And from all this, we found the T5 Stork title is a generative model which produces salient text to predict a document title is best performing. And also somewhere here in the long document, the generative models are fine-tuned on in-domain documents. So if you basically, the point of all of that is if you use domain knowledge to train your system, so if you use pre-trained language models, which are basically using domain expertise, the results are better. So the takeaways that we have from the Gen Compare sum is that it is an unsupervised method, which is quite important for, you don't need lots of training data. And this can be extended to any length of document. It does not require a corpus of annotated training data. It performs with both strong supervised and unsupervised baselines for long and short documents. And from all these methods, the best basically performing model uses the title and the document pairs for the generative task. So basically, this is what is the salience. And this actually can be, this type of information, title and document pairs, you can find it in absolutely any domain. You have papers because you have a title and a document. So you don't need to have manual labeling task effort. So another interesting basically evaluation includes human evaluation, which actually we're currently very active in engaging with different users. And this is actually also, I would be very interested in working with teams to support the automatic generation of our summaries and to do a human evaluation on other data sets, for instance, clinical notes, medical conversations, etc. I'll come back towards the end to tell you about the evaluation, because the metrics, besides the automatic, is actually how a summary is coherent, how a summary is readable, if the summary is factual. And this, it's a very interesting work that needs cooperation between the computer scientists and the users. So of course, there are many other directions that we can do, like selecting for salience. For instance, what we call specific zones in the document. You might want to focus on for instance, conclusions or methods or, discussions. So there are different types of sections in the document that actually could be very interesting. Now I come to something which is very interesting, I think, for you. How am I doing with time? Sorry, because I don't have any, I hope.
It's 1:45 now.
So how long do I have? A quarter of an hour?
Like maybe another, well, if we could leave about 5 to 10 minutes for questions.
Okay, so and now I'm going back to Pico, obviously, because this is your interest. So now we tried what we did also. We incorporated medical knowledge to these pre-trained language models to improve extractive summarization. And the reason basically we are doing that is because if you do direct fine-tuning of the PLMs, you cannot capture medical tasks. And this is the same idea as with the Pico prioritization. You can, in PLMs, they don't consider domain knowledge, they don't consider entities, they don't consider technical terms. And typically, they're picking up sentences which have high frequency, but those are not informative. So the whole idea is to infuse medical knowledge into our models to improve extractive summarization. But as you can imagine, this is important for other tasks as well. You know all that, so I'll skip it. So how we do that, our method is to integrate, as I said, the medical knowledge. We used, I'll just try to make it a bit simpler, what we call a knowledge enriched pre-trained language model. So this model injects the medical knowledge into the pre-trained language model using Pico annotations. And to do that, we use what we call a knowledge adapter. Now, a knowledge adapter is something, it's a kind of a lightweight training framework to be able to learn from this information. Now, this framework uses both generator and a discriminator. Now, the generators we have in deep learning, we reconstructs what we call the missing tokens in an input. When you have an input sentence, you have missing tokens, and discriminator makes the predicts the Pico elements for each token in the input sentence. So these train adapters capture knowledge and basically they are infused into the pre-trained language model. So the reason we, I know perhaps if it's not your field, it's a bit, sounds slightly difficult, but the pre-trained language models are really guided by the knowledge adapter, and they're doing that during the fine-tuning. So you can have better relationships between the medical concepts. It's also, the important part here is it helps us to understand the global meaning of the document and to select salient sentences which contain the important parts, for instance, here in the Pico. So again, you're doing summarization, but our model, our PML has a knowledge adapter based on Pico. So this is the important part. I'll skip that because it's too, maybe, and this is about adapters, and I think I mentioned that, but perhaps it's, oops. So I'll go to the experiment because just for the sake of time. So again, we used the 4 biomedical literature data sets, PubMed Long, Short, Cordis for COVID, and Stork, which is again from Alan AI lab. And we compared the state-of-the-art extractive summarization methods and all those different, if you were not in the field, they might not tell you much. But for instance, Assessor is a Pico-guided evidence summarization method. All are state-of-the-art. So here we're looking at the different training things of the data sets. Oops. And here are the results. And again, I know they look a bit too much, but perhaps you can look towards the end about and BioBERT and PubMed all full. So we see that the framework based on the PubMed BERT model, which is biomedical, achieves the best performance. And I think, and the reason this is happening is because of the ability of our model to exploit the medical evidence using, in this case, the PICO elements, and also using annotations we call with the knowledge adapter. So the knowledge adapter in the model infuse specific type salient model, which could be anything, into the systems, which actually are typically black boxes. And that allows us to distinguish for our summarization the salient with the non-salient sentences. Now, why this is important and why we think the knowledge adapters are very important is because it improves the interpretability as well. So we know we have infused specific types of information to our models, which typically they're black box, to capture also the domain knowledge of any area. So basically, I think this is an important part to maintain here. And it's actually, the takeaways is uses a knowledge adapter to incorporate fine-trained medical knowledge into the PLMs. Because they're absolutely important. So even if you use a PLM for biomedical, you still have to use more information to make your summarization process more salient. And we looked at the different data sets, and we see also the ability to incorporate PICO in this case, because this is a kind of, we mentioned PICO from the beginning. The last part, and then I'll finish, and that's very important, is how we evaluate the summaries of biomedical text. So we have two types. We have, I mentioned rules. I'm not going to mention all the different BERT score, etc. Are the intrinsic evaluation. They allow basically, those metrics are limited to shallow lexical overlaps. So they're they're actually not sufficient to evaluate the summary. And if we are thinking about in systematic reviews, the synthesis, this is really quite important. How we need to look at other elements like paraphrasing, like terminology, when we look at similarities. And another part which is very important is the factuality of the summary, to what extent what we create is actually meaningful. And then the readability, which is really very important. And the readability is to what extent a document, a summary we have created, can be understood by people, and it doesn't have redundancies, doesn't incorporate false elements, etc. Factuality and readability are very important, and the human evaluators are helping us with this type of tasks because they can look at fluency, how fluent and coherent the summaries are, the interpretability. So very often, especially for domain-specific areas for summaries, I can see a summary and I think this is, and actually I tried to do with physics summaries and I thought everything was great, but then domain experts are saying, well, this shouldn't have been exactly like that. So if you have it for domain expertise, you need to understand the domain experts to understand, but also to see if they're factual, and the grammaticality, which is actually slightly easier. So as I said, the most summarization systems, we have lots of jargon and complex language, and all this actually affect the understanding. So we need to also create technical summaries, but also summaries that can be used by people, and they can be understandable by people. But the most important part is we can actually gear the summaries to readers' levels of expertise. So it's not just for lay people. You might want to generate a summary which looks at specific areas of interest of the user. So if you have a huge document collection, is not only how we understand, but we might, for instance, think about summarizing everything on interventions. And that's actually, I think, a quite important part of the summary or the automatic summarization systems. So I don't know, I think I will skip some of those, and I will skip the metrics. So I will skip those. Basically, maybe I will start, I had more slides about the long, but I'll skip all that because it's so. Perhaps I would say I will conclude now, and that's the time to finish my talk to allow you to ask some questions. So we have here their conclusions of the future directions. We think there's still a lot of work to be done, but as you have seen, how we create summaries which are incorporate their geared towards the interest of users is very important. And also the problem we have here, we don't have large scale biomedical data sets for text summarization. More efforts have to be devoted into this. We can use unsupervised or, for instance, distance supervised automatic annotations to do that, or even, for instance, federated learning to allow the development of models while we keep actually training data on the private side. Also, more efforts should be proposed to explore the unsupervised, we call techniques. I mean, we're doing a supervised, but this is more required, like how we can use data augmentation techniques for biomedical summarization. So another thing I mentioned about long documents, and people don't realize, but the transformers and the architectures are very constraining. So they have models like long-former, for instance, which are directly applied to these methods, but have they're quite limited. And we want to, as we have been doing, to investigate the most effective way for pre-trained language models in the biomedical domain to encode the full content of text. So that's another important. I mentioned a lot about users' preferences, and I cannot emphasize that more. We need more efforts to what we call controllable summarization of biomedical. By controllable is based on, the user's aspects and perspectives. For instance, methods to control several attributes is like the length or the readability, the text style, etc. And then we need benchmarks because there are not many currently. Okay, about the knowledge, and we're talking about knowledge infusion, more knowledge-aware models are needed. We worked on that, on infusing knowledge into our models, because we think a lot on kind of more neuro-symbolic approaches. So we want to incorporate domain-specific knowledge, like, for instance, UMLS, etc., to improve the summarization generation. And also the different varieties of types in biomedicine. So it's not just full papers, it's controlled, randomized controlled trials, electronic health records, scientific papers, etc. So it's important to also, when people are using benchmarks, to have a unified benchmark to support the development and the fair evaluation as well of the proposed methods. And last but not least, talking a lot about multimodality. Currently, I only mentioned textual resources, but very often in medical text, we have images and text. So we think about summarization that does multimodal summarization. Thank you for your time.
Thank you, Sophia.
Thank you. Oh,
Yeah.
that's an hour. Sorry about that.
Yeah, no, that's okay. You were able to cover quite a lot in that hour. I know that some folks probably need to leave. So if there's anybody who wants a burning question, who has to go, I'll let you go first. Okay, nobody.
Feel free to email me if you need any information. And that's actually as an opportunity. I know it's a bit technical, but for us and for me, it's important to work with teams like yours, working on systematic reviews, because we work so much on summarization. We're computer scientists, but we very much work with users, and the human evaluation and how we work with domain experts to co-develop our systems is extremely important. So I very much hope that my talk is a kind of triggers this interest of collaboration with your team, because I would very much like to follow up properly, doing some work together and collaborating on the evaluation of our systems. So the human evaluation is extremely important now in our field. And there's also emphasis, there is also possibilities for co-authoring papers.
Yeah, and I think that's one of the particular points that I noted is that, we do have more of that domain expertise, and certainly an interest in sort of the fidelity of what the summaries are with the main documents from a clinical perspective, but even creating lay summaries for other kinds of users.
So, actually, we are developing, we call them community shared task as part of a big conference. We're organizing a workshop, biomedical and NLP for the past 20 years. But part of this is we create a community shared task for biomedical summarization. And one of the topics is human evaluation, readability that I mentioned, and the other is lay summarization. And for this, there are so many things that we can only do with domain experts. So it's an open invitation to work with you and to work with my team to look at the evaluation models we have done, and just work on the areas that you're currently working. So we can produce automatically the summaries for you, and then we can work together with evaluation. So I hope that will be of interest.
Yes, I definitely will be in touch after our call. Is there anybody else who has a question? Okay, I really want to thank you for your time. And I don't think it was too overly technical.
Great.
Yeah.
I'm very pleased. I'm very pleased about that. That's good. So thank you again for inviting, and you need a coffee, and I need a coffee too.
Yeah.
Okay, take care.
Yeah.
Bye-bye.
Okay, thanks.
Bye.
All right. Everybody else, we will have another speaker in the series later in March, but I will share those details. Thank you for attending.